Gavi. Thank you very much, Gavi. And it's a great pleasure to present and talk about genetics in the first International Psittacine Congress. I hope we're going to have many more coming in the next following years. So, as closing the ends of the talks and the plenaries of this Congress, I would like to give you some thought-provoking examples about how we should be moving beyond a simple headcounts in conservation as a whole parrot conservation community and leveraging the power of genetics, know that for making more conservation success stories. So normally I start my presentations talking about parrots and what parrots are. I don't think we need to do that for this audience this time. And we all know too well that parrots are one of the most threatened orders of birds. And most of these threats to parrots relate to growing human population size and anthropogenic factors, one kind or the other. For example, habitat loss, including industrial level agriculture, as you can see on this chart on the left, affecting more than a third of the global parrot species. Also logging, for example, or the illegal wildlife trade that we heard many talks during the Congress in the past few days. And on the right, unfortunately, prognosis is not too bright for the future. Our simulations show that predicted that human parrot conflicts in the future will actually uh, be more, and many species will be moving up on the red list of conservation status, which is unfortunately in this case it's a bad thing to moving up because it means that they're becoming more threatened on the IUCN red list. So the pressures don't just reduce the population size per se, but critically um, they can they can erode the very foundation of the ability to survive, which is the genetic health of these birds. And so this is the challenge I'm going to talk about in this plenary. I start with this image in Tambopata back in many years when I worked in Don's project in front of this beautiful clay lake. I had many hours to look at these birds and had many thoughts. We of course did lots of counting almost every day, identifying species, and of course like identifying parrots species is it's quite easy, I guess, especially with macaws. We counted things like relative abundance, and even we used some clever techniques from arrival, like group arrivals, to estimate some population size of the species. But all those times, I just really wanted to understand more about these populations, and just had questions like, are these the same birds coming back to the clay lake every day? Are they closely related, or are they just males, for example, or females and groups, mixed groups? And as Don already mentioned in his very first talk, many techniques, unfortunately, are not feasible for parrots. We also tried to misnet them and really failed miserably. But fortunately for genetics, it's really applicable for parrots. And if we think about conservation historically, it's focused more on demographics, no? That always the numbers, how many individuals are left, how many individuals we need to save, what's the population size we need to reach. But the genetics adds a crucial second dimension to this question. It answers vital questions like things that demographics just could not answer by itself. For example, is this population viable in the long term? Are we losing the adaptive potential of these birds that is needed to face the future challenges, as we saw in the previous slide, like changing habitats, diseases, or climate shifts? So with all these threats, just coming more and more in the future and in our present, it's never been more important to have a real evolutionary potential that's needed in our rapidly changing world for survival of these birds and populations. And also, I want to add that genetic data is no longer just a nice to have thing or a standalone project. It's also becoming more and more essential as a part of modern effective conservation planning for parrots. And of course, it's not only applicable for parrots. Like if you work not just on parrots, but in other species, I think many of you also do other species than parrots. It's of course also applicable conservation genetics, and it should be applied to other threatened species as well. So let's first open up our textbooks and try to define some key concepts. I try to be quick and efficient. I think the first, and many of you probably already know about what genetic diversity is, but let's just define that this is the extent of genetic variation in a population or species or across different species. So just think about it like a currency for adaptation. And as geneticists, we measure these things with the so-called heterozygosity, which is basically just having two different alleles or versions of a gene. Or in other words, you can think of it as an individual is heterozygous for a certain trait if they have inherited one allele from their mother and a different one from their father. So they have already an inherited diversity at that allele. So genetic diversity is really generated over time by mutations. So mutations are creating new alleles, but it's a very, very long time. It happens over millions of years. And on this figure in the top, you can see that each colored dot represents a different allele. So that's our diversity that happens over time. And mixing different alleles from parents, that also creates new combination of alleles in the offsprings. So that also just contributes to more diversity in the population. But unfortunately, genetic diversity can and will be lost when habitat loss happens. For example, when it divides population, as you see under with those rhinos images. And just by chance, some of these remnant population will end up one handful of alleles, and another population will have different alleles. And if they stop mixing and mingling with each other, then this genetic diversity won't keep up. And it's very important, and this is basically the whole point of creating protected areas and corridors where individuals can move freely from different populations and rejoin, migrate, and spread their genes. It's really amazing, like how can just one single individual migrating from one population to another can contribute so much to keep up the genetic diversity of that population. It can really help a species to recover and maintain this diversity. So why is this important for you, for as a practitioner, for example? If you have a genetically diverse population, it's more likely that you have individuals who can survive a disease outbreak, for example, that they can adapt to changing food availability or tolerate shifting climate conditions or different habitats. So put it this way, genetic diversity is really our primary measure of population's resilience. So what happens when a population gets really small uh, and isolated? So this is quite a common scenario, I think, for many threatened parrots, as we know quite well, when related individuals are more likely to breed. And this is going to lead to inbreeding. On the image on the top, you can see this is a predicted decline in heterozygosity that we already defined over time, over generations. So you can see if this really depends on the population size. So if you have a large population here, for example, 200 individual, this diversity declines really slowly. But if you have a really small population like 10, you can see that it declines really rapidly and drastically. And so usually we measure this by a so-called inbreeding coefficients, which is just an inverse measure and calculated from this and marked with F. And you can also see that the same thing applies with very small populations. This inbreeding coefficients grows very fast. So this leads to a so-called inbreeding depression, which has a very negative effect to our conservation outcome and our conservation work. And you don't need to think about that your bird's going to get depressed, not that kind of depression, but this is manifesting to, for example, decreased clutch sizes or poor fertilization rates, high chick mortality, for example, or compromised immune systems. So it has a really long-lasting effect, and it's really hard to turn back. So this is a very critical factor that can undermine our work, and also in captive breeding and reintroduction success if not managed well. And as we saw, for example, yesterday in Ugostock with the Spix's macaw, that was a group that really well managed for down to every single individual. So, which is a great example of, what you can achieve just a few birds if you manage the population genetically quite well and know which ones to pair up. So the other extreme, which Renato just talked about a few minutes ago, is the outbreeding depression. And this happens when mixing populations that have been isolated for a long time. And this can lead to a different kind of depression that's on the outbreeding depression. Here the consequence we call introgression, which is basically a flow of alleles from one species or subspecies to another one. And that's basically threats the genetic integrity of that given species of questions. So in outbreeding depression, it happens when, for example, a locally adapted gene complex is broken up by this other bird that's not adapted to that environment. And the resulting in offsprings that are much less fit for either of the parent environments. So that means that this will have a much lower survival, but if they do survive and spread these broken up complexes in that population, it actually eventually leads to genetic swamping. And that's not good for the wild population. So we can also face challenges like hybridization, and they usually call that when two different species meet with each other. And also, for example, this happens between native and introduced parrot species, or it can happen in captivity as well if it's not managed well. So that's good that we have genetic tools, which are essential in this case to identify these situations. And this is, for example, just one of our recent work where we tested um, in a breeding program military macaws and great green macaws as one cohort. And you can see it clearly that we could identify the hybrid species, the hybrid individuals between the two species quite well. So this can be important to consider, for example, for release or relocation programs as well to prevent unintended harm to the population that we actually want to save. And one more thing, one more key concept is the population genetics. When comparing the genetics of a single or isolated population, that can also help us determine different units, conservation or management units. So let's see this example on the right side, where we have alleles in a large versus fragmented populations over time. What happens here? So we start with four different alleles. These are our genetic diversity, let's call four different alleles. And with time, we have, in a short term, we had an isolated population that just by chance they can retain some of these alleles, but they will go into fixation. So let's just call A1 and 2 and 4. They will get fixated in those small populations. But if you take it an overall diversity, they might actually retain a much larger genetic diversity than the single population, the single large population that we have, that just by chance it can lose one of these alleles. But as these small populations, the isolated ones, they become more and more inbred, then eventually they will go extinct, some of them, just by chance, by stochastic event or because of inbreeding. And they actually ended up losing more diversity than the large population itself. And without testing these genetically, this might happen unnoticed or unexplained, but active management can actually help to retain this diversity. And this is like the underlying idea of why we need to keep a population large to maintain the genetic diversity over a long time. And if we have already isolated populations, why it's important to get them connected. And this is also true for captive breeding. If we're managing, for example, a threatened species in different zoos that would represent these isolated populations, it's really important to also mix these different zoo populations with each other, at least a few birds, that you can maintain this diversity over a long time. And so once we know a population well, like we analyze some samples, we have some information of the population genetics, it can also be used to create, for example, a genetic map and assign provenance of different individuals of unknown origin. For example, if you give me a sample from one of these, like, for example, the A3 population, and we analyze and we know about the different other populations, we could probably assign with high certainty that it was coming from that population just based on the genetic information we already know about those populations. But these techniques can also be used to reveal taxonomic uncertainties over the large scale, for example, define subspecies boundaries, or even discover cryptic species in over large scale or in one population. All right, so I think enough for the textbook definition. I think we all know now why genetics is important. Let's go to the lab. And the good news is that our toolkit is more powerful and accessible than ever before. You, as parrot researchers, you can capitalize on a huge advantage of parrots, that their blood contains nucleated red blood cells, as all birds and reptiles does actually. This means an amazing DNA source in blood. So you just have a few drops of blood, and that's enough to do almost like a whole reference genome for your species. But of course, if you don't handle birds, you can also collect feathers, eggshells, swab samples, or even degraded DNA of museum specimens are really, really valuable, and you can extract DNA, even trace amount of DNA these days. So this means that we can gather vast amount of data without even capturing or stressing the birds. And so normally once the sample reaches the lab, the pipeline needs to do a DNA extraction, but this can even happen these days in the field without electricity with new technology. And then hopefully then we do a quality control and then some kind of analysis. And still the most widespread analysis still today is the PCR, where we amplify a single fragment of interest. And then we can visualize this with GL electrophoresis. And here, for example, on the right, you can see just a simple sexing image where two lines means this is a female and one line is a male. So this is a very simple but really brilliant way to analyze a DNA fragment. But if we are interested into a genome-wide diversity testing, we need to get some genetic markers, and that's a bit more complicated. The old style markers, we call them the microsatellites or short tandem repeats, where the diversity, these are like parts of the non-coding part of the genome, so that we call it just junk DNA. And the diversity here is, or differentiation is by the number of the repeats that each individual has in different parts of the genome. So using enough of these markers, it's still very, very powerful to identify and species or individuals and separate them from each other. And this is the gold standard, for example, because of this still in forensic analysis and human forensic as well. But with the genomic technology, we phasing more into next generation sequencing and single nucleotide polymorphisms or SNPs that are just basically detecting one single change in a part of the genetic material, or ACT or genome, one of the nucleotides change. And this could be in a coding or non-coding region. If you have a good reference genome with good annotated reference genome, you can even decipher what that change is, what that change means. For example, if it's coding for, let's say, for a protein that's important for adapting to a more warmer environment or something, you can also know that. But of course, it's not going to be coded by a single change. It's of course many changes, but we use thousands or tens of thousands of these SNP markers when we look at a genome of an individual. So we moved from a single markers to genomics, and this allows us to assess the entire genetic landscape of a population, and very importantly, much faster and cheaper than before. And this makes genetic monitoring a really practical reality to field-based conservation programs these days. And so we talked a little bit about sequencing. I just quickly want to run through the evolution of the sequencers we have. So the first generation sequencing, or called it Sanger sequencing, from the end of the 70s. This was the first gold standard for sequencing DNA, and it's actually still a gold standard because it's really accurate. It's almost 100% accurate. So if you need to sequence some part of the genome, it's really useful still. But it's not cheap, and unfortunately, it's quite slow. So the throughput is very low. They sequence is only one DNA fragment at a time, or depends on how many capillaries you have in the sequencer. But while we did the first human genome sequence this in a few decades, and after many billions of dollars spent on it, now, of course, with the big revolution of the next generation sequencing, or NGS, or we call it second generation sequencing from the 2000s, it was a really huge revolutionary jump in sequencing. Because also this a short read sequencing, because it just sequences up to like 300 base pairs, so it's like really small pieces of DNA. But the big jump here is the massively parallel feature of this. So it just generates a huge amount of data in very short time. So now that took, like decades to sequence a human genome, you can do it in a few hours. But the issue here is that it generates these short sequences. So you can imagine this jigsaw puzzle, a really tiny, small jigsaw, and a huge image that you need to build up. So if you don't have a reference genome, it's really hard to build up the whole picture from de novo. And we didn't have many reference genome or at all at the beginning. Now we have more and more, but this technique is usually used when you already have some reference, and you can do like a resequencing. And so that takes us to the third generation sequencing, the newest one. And we also call it long read sequencing, because it can sequence much bigger, like thousands to millions of base pairs, like much bigger chunks of DNA. So you can imagine all of a sudden your jigsaw puzzle got really big pieces. So it's much easier to build up the picture of the whole genome. And these are platforms like PacBio or Oxford Nanopore technologies. They came with the lower accuracy, but now it's much more accurate. And actually, PacBio is becoming the gold standard for reference genome production. So if you need to produce a reference genome, that's one of that one combined with an Illumina is the gold standard these days. And Oxford Nanopore is they also rolled out with this really tiny sequencer that you can see in this image, where you can actually take it. It's like a pen drive size sequencer that you can just take it to the field as well, plug it into your computer, and then you can just sequence it in real time, your whole genome, or part of it that you're interested to sequence. All right, so let's take this to the field for real and see what genetics can actually help us in parrot conservation. So I think the most obvious thing that genetics can help is taxonomy and phylogeny. It really helps to resolve these taxonomic uncertainties that existed or still exist in some species. And it's important to keep in mind that the IUCN Red List, for example, as many other, like national red list, they handle species as the main unit of conservation. So it's really important to know, what a species is, and what's a subspecies, or define, whether a subspecies is actually a species, because then it gets a different conservation status. But it's also important to look much deeper into that, the intraspecific resolution of, of the species, and that also can see some management units. For example, there's so many examples for this, but I just took one with the African gray parrot, where they showed that these populations in the Gulf of Guinea Islands, they have an evolutionary significant unit because they genetically different from the mainland populations, but not too different enough to call it as a subspecies. And they also under heavy poaching pressure and having these really different genetics. They argued that it's an evolutionary significant unit, so it's really important to conserve these populations, even though it's not a different subspecies or a different species of the gray parrots. So as I mentioned, species identification is quite easy for parrots, but it's not so easy when they still inside the egg. And that happens. For example, this is a case study from Australia when eggs were confiscated at the airport, and unfortunately, because of the quarantine procedure, they had to also autoclave these eggs. So there was no time to hatch the eggs, and then the law enforcement officer tried to hand raise them and see what will hatch from those eggs, and then identify the parrots. But they took instead DNA samples, sequenced some part of the mitochondrial DNA, and they were able to identify that they were actually parrot eggs. And this was followed by a court case that they presented this as an evidence, and the smuggler was prosecuted. So this is a positive story. This is another story from Brazil. It's also very, very similar. Egg smuggling, they use mitochondrial DNA to also show that these were parrot embryos inside those eggs. But unfortunately, this had a different outcome. The results were not used in the police investigation, just presented as like a scientific study. Uh, and unfortunately, this is not just Brazil, but many, many other countries. And I think Australia is more like an exception than the rule. And these cases, like wildlife cases, they're still not treated very rigorously and not taken much importance to take it to court. So that's why it was really interesting to talk, for example, to Carlos yesterday about the Lares Macaw identification project that he mentioned. That was the first case in Brazil where they actually use these evidence for law enforcement purposes and investigation. So, and hopefully that's going to be more like the case in the future in not just Brazil, but in also many other countries to follow this example. So this is another interesting case study from Australia to show how genetics can also support some law enforcement investigations. So some people spotted some suspicious activity in a reserve. Some guy was climbing a tree and removed something from a nest. So people called the rangers. They came out. They checked the nest and they could only find some eggshells inside, which they took out as evidence. The people who reported this guy also took a picture of the car that he used, and based on the plate, the police could find this guy's home, and they went out, they visited him, and turns out that the guy was actually a parrot breeder. And they find the red-tailed black cockatoo, but just a chick, and it's a bit hard to raise a chick without parents. So or breed this species without parents. So that was also very suspicious. They took samples from the chick and they did an STR typing that completely matched the eggshells recovered from the nest. So that actually placed this individual to the crime scene. And there was a criminal conviction followed this, and the guy was actually fined with $1,000, and he had to pay for court costs. So I think this is again an interesting example just how you can really use genetics and human forensic techniques the same way as you would do in a human forensic case, but apply to wildlife and especially parrots in this case. So now let's go back to Peru. This is the famous Peruvian Amazon in Tambopata, the in front of the clay lake that I spent so many hours watching. So we ended up eventually collecting feather samples non-invasively under the clay lakes, and then took them back to the lab. And based on the whole genome of the scarlet macaws, we actually developed some STR markers, and that helped us to individual genotype all these feathers. So they actually we were able to identify, not just the amount of feathers that we had, but just filtered out how many individuals that actually represent, because they can molt more than one feathers at the clay lick. And we did this collection regularly, like every 2 weeks or every month. And so that was really interesting, because we could identify just based on the feathers and genetics what sex they had. And so it helped us do some demography study. For example, most of the individuals that visited the clay lick in the breeding season, that was they were males and closely related. But we were also able to identify subpopulations over the landscape, and also down to individual level. As I was, talking earlier about the STR markers advantage to be able to identify individuals, not just, groups or populations. We could see like recover genotypes or feathers from the same individual from different parts of the landscape, from different clay licks, and that gave us some information about their range or their movement. And because we did this regularly collecting, this was kind of a genetic capture, mark recapture study, where you basically, you see how many new feathers you get into your collection genetically in at every collection time, that that can help you to estimate the population size, or at least of that clay lick. And that was also interesting to calculate that and see that these different clay licks had different clientures. They not just mixed randomly, but usually the macaws using one clay lick, they often come back to the same clay lick and use that. So they're more related individuals for using that over time. And we also have now a long-term project in Australia on the now critically endangered swift parrots, led by the group of Dee Stojanovic and Rob Heinzen. This is a nomadic and migratory small little parrot. It's really hard to capture as well, but we know where they breed in Tasmania, and it's easy to get samples from the nest, from the chicks. And over many, many years and many, many samples, we showed some genetic evidence of some severe extinction risk. And not just that, but the team also found that there's a predator, a sugar glider, that killing the breeding females and the eggs. That also led to a sex bias in the population. So there's many more males, adult males. And we also showed genetic evidence of polygamy, very strong polygamy that reduced the population viability. And eventually, this species got uplisted from endangered to critically endangered now, not just because the genetics, but the whole project. And interestingly, we were also able to calculate the population size, in this case, just purely from genetic samples, from the effective population size calculations. And it was much lower than any of the previous estimations like BirdLife had before. So that was really intriguing, but really mirroring population viability analysis result that was done a few years before. And this is the year when we did the genetic study, and the population size that was predicted is actually matched the population size, just a few hundred birds instead of a few thousand birds of the present. But unfortunately, the species is still plummeting towards extinction now. So that rang many bells, and the government as well. And they thought we need to think about to establish a backup population. And also, there's already one population existing in zoos in Australia, but they were not established for conservation and not managed as conservations like Ugo's macaws. And we had lots of unknown birds, like birds with unknown origin and pedigrees. So they asked us in 2024, when a formal management started, and they wanted to maintain the genetic diversity of this species in captivity, to actually sequence the birds in captivity and see what we can tell about them. And unfortunately, we couldn't tell, very good stories about them. They're really highly inbred. You see, they're very different from the white population, which is green. But there are few individuals that matched with the population that could be useful, but that would not be enough to actually start a whole funding population from scratch. So we also did some calculations based on the genetic information that we had available from the wild as well in the many years of sampling. And so we estimated that what is the how many individuals we need to have to maintain the 95% of the heterozygosity currently found in the wild population. And we had this number estimated to be around 11 individuals. But of course, it's not that easy. Like it needs to be, 11 individuals that can actually breed with each other. We need to consider males and females. Uh, but at least this gives some guidelines to this workshop that we participated in, and where the government and the management will need to decide some important things about how to manage this species and how to establish a funder population in captivity. But genetics cannot solve this problem alone. And as you see in the image above, they still clearing huge amount of forest, key habitats for the swift parrots, that there's no place where they can go back and nest. So it's really it's just one part of the conservation toolkit. You also need to be done much more to save the species, to have a place, for example, where we can release them later on. And I also talked a bit about this sugar glider. This is a nocturnal marsupial that is native to Australia. So that's why it's protected also in Tasmania, because it's still part of Australia. Uh, and this is the swift parrot killer. So this is one of the main predator. That's the main reason a part of the forest clearing why the species is declining. And there was some suspicions that this might be introduced to Tasmania. So there was this study done that's really clever. They just did sequence 3 genes of of this species, the sugar glider, and they found, based on museum specimens and samples collected from Tasmania, that the closest relative from Victoria, from mainland Australia here, the difference genetically between these two populations are around 150 years. But geologically, Tasmania split from Australia around 12,000 years ago. So this was a clear evidence that that was a human-mediated introduction to Tasmania. And from now on, the eradication is legally possible, because now it's not protected in Tasmania anymore, just on mainland Australia. And lastly, I just want to give one more example for how genetics and forensic genomics can be used to fight the illegal wildlife trade. This is actually my current research. For now, we only have just a handful of countries and species we focus on, the most heavily traded species that we try to focus in: Indonesia, Solomon Islands, and Mexico. We're sampling from the wild and from the trade. And from the wild, we're building a genomic database of these species that can serve as a reference for later on to check the samples taken from the trade. And also this would be available for law enforcement authorities to keep building it, and also to consult if there is a confiscation of that same species. So this is very similar to what already exists for humans, but there's not like a standard database for parrots yet. Also, this is you see a QR code up in the corner. If you're interested, or you have one of your study species is really threatened by trade, and you have access samples in the wild, and you're interested to join us, just there's a form that you can fill out, and we can talk if you're interested. The idea is to identify the provenance and to map the trade that can help law enforcement agents to focus on, once we know where things are coming and where things are going, on the trade and highlight this hotspot. But also it would help the release, because in many cases, for example, in Indonesia, the big stopping point is that they don't know where they came from, and that's why they don't let us release them to that location, to the correct location. And so if we have a toolkit and a really vetted database that we could show that these birds were coming from that region or this island, then hopefully it would have a much better collaboration with these authorities as well for release. And time is really key in after confiscation to get the birds back to their habitat. So that's why we're also developing an in situ sequencing technology and capacity building, local capacity building, so they won't rely on on foreigners, or they don't need to send the samples to Australia or the US to get the results back in a few months, but they can actually do their in situ eventually and check the database themselves. So that would be quite crucial and time saving, which is also very important in conservation. And also, since we have the birds in hand, we also do some disease testing, just using metagenomic techniques. And yeah, I think that's mainly it. So let's just summarize it and wrap it up. So in summary, a genetic perspective can really transform our work in a big time. It lets us look under the hood of the populations we manage, and it really helps us to diagnose problems that already exist, for example, in breeding, and also avoid creating new ones like outbreeding, also to manage our captive assurance populations effectively. And it can also provide forensic power to combat the illegal wildlife trade, as you saw. And it adds a layer of quantifiable measurement of conservation actions. So my call of action to you all, I would really like to encourage all of you, and I know many of you already do this, to champion the inclusion of genetic analysis from your project planning all the way, from funding proposal to management. Uh, Non-invasive sample collection is really easy. If you handle birds, you can also get some blood samples, which is best, but even just a few feathers is really important. If you already have access to that, just save them, keep them for later DNA analysis. The costs are really dropping, and but it really depends on what type of samples you have, what amount of samples you want, what questions, techniques, etc. But don't get that it's going to be too hard and too expensive. Don't get that into your way. I really encourage you to foster collaborations with geneticists to select the best and most affordable techniques that can answer your questions. And it's really hard to give like just a per sample cost right now, how much it would cost. I'm sure you have these questions already now in mind, but maybe it's something to go deeper into a conservation genetics workshop next time in the next conference. But I can say, like, for us geneticists, the most complicated is to get permission for sample collection. So I can start swearing about hours how bureaucracy gets into our way and what we need to master just to get a single country permission. If you already have this sorted and you have a project and would like to include your species into a wildlife trade database, reach out to us. But of course, if you have other priorities, other questions to your study system, you can also reach out to other many geneticists. We have really good people here in the Congress, Renato Caparros, Dr. Escalante, I think the contacts are there, or we can put you in contact with someone in your local area or university. And I think that's all. I gave a few examples, but if you want to read some more examples, we have a review on all things parrot genetics in an open access journal. And yeah, at the end, I just really want to urge everyone to work together and make these tools as a standard part of many parrot conservationist toolkit. And let's show an example to follow to conservationists working on other taxa. I think that would be really cool to showcase. And with that, thank you very much for your attention.
Thank you, George. That was a really